Hey everybody. So in this video we're looking at sex-linked genes. So far we've only looked at genes that are found on the autosomes in a cell, so the normal chromosomes. And this type of inheritance is called autosomal inheritance. And so all the Punnett squares and inheritance that we've been learning so far applies just to these genes, which is where most of genes are found. Because remember, we have, in humans anyway, we have 44 autosomes and we only have 2 sex chromosomes. So the vast majority of genes are found on autosomes, so the inheritance pattern we've learnt so far applies. But some genes, obviously, are found on the 2 sex chromosomes. And these types of genes have a slightly different way that they're inherited. And the reason for that is because there's 2 different types of sex chromosomes. As we know, there's X and Y are the 2 different sex chromosomes. And we know that for females have 2 X chromosomes and males have only 1 X and then 1 Y. And it's really this difference that means sex-linked genes are inherited a little bit differently to normal. So when we hear the name sex-linked gene, that basically means a gene found on the sex chromosome. And generally it means found on the X chromosome. If it's a gene on the Y chromosome, we specifically call that Y-linked. So if you ever hear sex-linked, we're normally talking about X. Okay, so how are these genes inherited differently? Well, let's use an example. So this is a really common sex-linked trait in humans, and it's colorblindness. So the gene that controls colorblindness is found on the X chromosome in humans. So somewhere on the X chromosome is this gene that makes people colorblind. Well, that can, if it's faulty. And here's what colorblindness is. So it makes it really difficult to see the difference between reds and oranges compared to greens. So most of us would see peas and carrots like this. People who are colorblind can't see the difference. And these kind of tests are pretty much the way that doctors diagnose colorblindness. So most of us can see the number 74 in here in green, but people who are colorblind couldn't tell the difference between those colors. So colorblindness is X-linked, it's on the X chromosome, and it's recessive. So we can still have dominant and recessive sex-linked genes. Can still be dominant and recessive. Well, the phenotypes can still be dominant or recessive. Okay, so with colorblindness and with all sex-linked genes, we write their alleles slightly differently because we need to show which chromosome that allele is on. So for X-linked genes, we always write X and then the allele up in superscript at the top here. So for colorblindness, we'll use the letter C. Actually, I'll use B. It's easier to tell the difference. So an allele that's on the X chromosome that's dominant, we'd write like this with the capital B. And then if there's a chromosome that's an X chromosome and it has the recessive colorblindness gene, we'd write X with the small b at the top here. But then for males, they only have one X and their other chromosome is a Y. So we just write that they have a Y chromosome. So that means if we're writing the genotype of a male, males are either going to have the dominant allele and a Y, or the recessive allele and a Y. Which means males only have one copy of this particular allele, which means they're never heterozygous. So for sex-linked genes, when we're talking about males, males are called hemizygous. Hemi meaning they only have one copy of that allele. So for males, it really doesn't matter about dominant or recessive. If they have the faulty allele, then they're going to be colorblind. And if they have the healthy allele, then they're going to be normal seeing. But for females, they still have two copies, so they still have either homozygous or heterozygous, like normal. Okay, so let's practice doing a Punnett square to show how some of these sex-linked genes can actually be inherited. So in the first example here, we've got a female, and she is going to be homozygous normal site. So she has X big B, X big B. And she's going to be having children with a male who is colorblind. So he has the colorblind allele and a Y. So he doesn't need two copies because the only allele he has is the colorblind one. Alright, let's set up our Punnett square. So for the male, he can give either his colorblind X chromosome or his Y chromosome. And the mother, well, she can only give her X chromosome with a healthy allele. So then the possible children they can have, we will follow through like a normal Punnett square, where each parent donates one of their chromosomes, because that's the possible gametes that can be made. X big B and Y. Okay, so the possible children that this couple can have. Well, firstly, we need to think about which children are male and female. So these two options are going to be boys, because they're the ones that receive the Y chromosome from their dad. And these two are girls. So for the boys, well, they're both the same here. The only option, if they have a boy for this couple, is to have an X with the healthy allele and a Y. So these boys are going to be normal seeing and obviously male. And then for the girls, well, they're both X big B, X little b. So they're heterozygous, but they're still normal seeing or healthy. So when we're writing the options here, the possible genotypes, well, we're going to have 50% X big B Y, and then the other 50% are the females, which are X big B, X little b. So they're going to be carriers because they're carrying an unhealthy allele, but they don't actually have colorblindness themselves. And the phenotypes, well, all these children are actually normal seeing. So we have 100% normal. Well, normal seeing. Okay. So you can see if there's a colorblind male and he has children with a female who is homozygous, then they actually aren't going to have any colorblind children. So let's look at the other option. And this would be if one of these children, these females who are a carrier, if they decide to have children. So the female here is a carrier. She's now the mother. And then if she has children with a normal seeing male, let's follow along what happens here. So the male can donate these two chromosomes, these two alleles in his gametes. And the female, she can donate these two alleles. So the first possible child is a girl who is normal seeing. The other possible child is a girl who's a carrier, still normal seeing though. The other possible child is a boy, he's normal seeing. But then this boy here, he will be colorblind because he's received the faulty allele from mum and he's received a Y from dad. So he's hemizygous and he only has the faulty allele, so he is colorblind. There is another way. So the only way to have a female who's colorblind is if the mum is a carrier and the dad has to be colorblind too. So he would have to be carrying a faulty allele. So with sex-linked genes, that means overall, a sex-linked trait is much more common to be seen in males. Because males only need that one copy of the faulty allele for them to have the condition. That's the final point to make with sex-linked genes. They're normally seen in males, they can be seen in females, but it's a lot rarer.